classmates. The following video is based on my Human Genome Project presentation. So we're going to start out with what the Human Genome Project is. The Human Genome Project was an international scientific research project with the goal to determine the base pairs that make up the human DNA and map out all of the genes of the human genome from both a physical and functional standpoint. So when did the Human Genome Project start and who created it? The Human Genome Project took 13 years to be completed. It was started in October of 1990 and it was completed in April of 2003 following the publishing in 2004. It started under the leadership of the American geneticist Francis Collins and then leading to scientists around the world to join. This was the first ever major collaboration of its kind and the largest biological research project ever undertaken. So who took part in this research? A total of 20 institutes from 6 different countries were a part of this research. Those countries included China, France, Japan, Germany, the United Kingdom, and the United States. Some of those institutes that were a part of this research were Baylor College of Medicine, Washington University School of Medicine, Wellcome Trust Sanger Institute, among other institutes. So how was the research conducted and projected down? So the research started off by sequencing technology with only a few hundred base pairs of DNA at a time. Then it had to be broken down into smaller pieces for sequencing, following with the first 200,000 base pairs section or clone. Then subsequently inserting it into bacteria DNA and then moving forward between collaborating institutes. After that, the clones were broken into smaller pieces, which were 4,000 to 6,000 pairs, and reinserted into the bacteria. They were cultured or cultivated, however you want to call it, to make enough DNA for sequencing. The bacteria clones were then transferred to tubes, sliced, and then DNA was removed. For this, they used the Sanger sequencing method, which was used then resulted in data being put together to form the whole genome sequence. So how was the research conducted? Here's a diagram explaining a little more of how that human genome was sequenced. Sorry. Here is the timeline of the HCP. So right after that, like I said, it was started in 1990, and the first gene to be mapped out was the BRCA1, which is a gene for breast cancer. In 1993, the first 5-year plan for the HCP was published, and Sanger Institute, which is the UK, joined. In 1994, the AGP Human Genetic Mapping Goal was achieved. Following with 1995, Genetic Privacy Act was passed, as well as the first bacterial genome was sequenced, which is the Haemophilus influenza. In 1996, the first human gene was published, or map was published, The yeast genome sequence was sequenced, and the HEP's mouse genetic mapping goal was achieved. Following with 1997, E. coli genome was sequenced and the sequencing center was established. Following 1998, second 5-year plan for the HEP was published. Japan's Genomic Therapy Center was established, and the genome of the roundworm Caenorhabditis elegans was sequenced. SNP sequencing was started, and the Chinese Human Genome Center was established in Beijing and Shanghai. In 1999, the sequencing of the chromosome 22 was completed and was published in Nature. Following with 2000, working draft of the human genome was complete, and the former U.S. President Clinton and the U.K. Prime Minister Blair supported the free access to genome information. In 2001, the working draft of the human genome sequence was published in the Nature and Science. In 2002, working draft of the mouse genome sequence was completed and published. And lastly, in 2003, the finished version of the human genome was completed. It was done, and they were done with all of their goals achieved. So, who has access to the Human Genome Project data? So, to put it in simple terms, everybody has access to the project data. It is provided with free open access to everyone, any of the scientific community and the general public, as well as public databases. They have access to more than 50 species of the genome. And lastly, what happened after the HEP was finished? After this was all done, um, they um, were officially done. The completion of this project led to many outcomes such as mapping, which they knew now number, location, size, and the sequence of a human genome is now like established. Screening this has allowed to produce specific genes, help to detect sufferers and carriers of genetic diseases such as cancer, sickle cell anemia, um, what else can I think of? Diabetes, anything that is hereditary. Medicine, the discovery of new proteins have led to improved treatments and make them better, more functionable. And lastly, the ancestry comparison or comparing other genomes has provided insight into the evolution and migratory pattern of mankind. Um, and yeah, that is my project. Hope you guys enjoyed it and there was enough information for you guys. And thank you so much.